So I'm assuming that you're hearing me well because I've seen some people respond on the chat. And yeah, so that's our title for today. And just a little bit of recapping on immunology concepts, general immunology concepts. We have several compartments within our immune system. Our immune system can be divided into innate immunity, adaptive immunity, and then you also have some other physical and physiological barriers that protect us from invading pathogens. So basically the role of the immune system is to protect you from any invader. The first protection that you get is from the physiological and anatomical barriers, like the skin, for instance. In the respiratory tract, you have the ciliary, the cilia that clear anything that is trying to access the lungs and push it out. And then, of course, in the stomach, you have the low pH, highly acidic environment that is able to eliminate things that might be trying to access the body through the gut. Things like tears and saliva that have enzymes that are able to eliminate some of the pathogens that might try to invade through those mucosal surfaces. And then, as I mentioned earlier, you have the innate immunity. And in addition to that, you have the adaptive immunity. And these are what we classically call the immune system. And the innate immunity is mainly composed of components such as cells and soluble factors that are always there, always ready to protect the body if something happens to try to access from outside. So this innate immunity, basically it doesn't need any preparation. The cells are ready, and these are cells like granulocytes, like like neutrophils, eosinophils. You also have things like macrophages, mast cells, dendritic cells, natural killer cells. As the name suggests, they are naturally ready. They don't need any preparation to be able to effect their immune activity, immune protection. You have things like complement. You have some antimicrobial peptides that are the soluble components of this innate arm of the immune system. So those ones will always be there. And then on the other side, you have the adaptive immunity. And as the name suggests, this adaptive immunity needs to get to be modified, so as to adjust to the nature of the invader. So in that one, in that arm, you have T cells and B cells. B cells act by producing antibodies, which end up giving us the soluble components of the adaptive immune system. So we're going to discuss a little bit more about how the adaptive immune system adapts, as the name suggests, to any invader, so as to be able to fight it more efficiently. And before we go further into that, I thought it would be nice to also revisit the organs that are important in the immune responses. And these organs, we call them the lymphoid organs. And so we have what we call the primary lymphoid organs and the secondary lymphoid organs. So the primary lymphoid organs, these are places where lymphocytes are generated. As the name suggests, lymphoid organ, which comes from the word lymphocytes. And lymphocytes are some of the major components of the immune system, like T cells and B cells are usually called lymphocytes. So basically, the primary lymphoid organs is where lymphocytes are generated. And for T cells, is the thymus, and the B cells are generated in the bone marrow. And then you have the secondary lymphoid organs. These are places where lymphocytes mature. These are the places where immune responses take place. And this is where lymphocytes get educated, if they are the type that usually require education. And those are the same organs where activation of lymphocytes takes place. So the secondary lymphoid organs will mainly be things like the lymph nodes, which is the one that we study most, and which really plays a major role in generating immune responses. But then you also have things like the spleen, the Peyer's patches in the small intestine. Yeah, so you basically you will have different versions of lymph nodes or lymph node-like structures, even things like tonsils, which have very close similarity to lymph nodes, but they are actually functionally grouped together with Peyer's patches because they are part of the mucosal compartment. So those are what we call the secondary lymphoid organs. So primary lymphoid organs being thymus for T cells, the bone marrow for B cells. Then you have secondary lymphoid organs that are the places where immune responses mainly take place, and that's where immune cells get activated and get educated to fight the invading pathogen. So this is another presentation of this innate versus adaptive immune system, but it is showing another a slightly different piece of information. So remember, we talked about the innate immunity, which is always ready to fight. It doesn't need to be educated. These are things like natural killer cells or granulocytes or dendritic cells or macrophages, or even soluble components like complement. Just as a way of recapping that. And then on the adaptive arm, you have the B cells and T cells. And of course, the B cells can produce antibodies. T cells can be subdivided into CD4 T cells versus CD8 T cells, depending on their function. But then the reason why I'm showing this is to show that there's a slight overlap somewhere there, where you have some cells that have features of both adaptive immune cells and innate immune cells. And these are cells like gamma delta T cells and natural killer T cells. So these natural killer T cells are very similar to T cells. At the same time, they also have similarities to natural killer cells, like this one. While the gamma delta T cells are T cells that have another different kind of receptors on their surface. And those receptors look fairly conserved. They are not as diverse as what we see in the T cells. And those are the receptors that are important for the mediation of protection that these cells are able to mediate. So because of the relative conservation of those receptors in these T cells, it means that they don't need a lot of selection, and they also have some other features of innate cells, but mainly the conservation is the one that makes them fall into that. So they are there in between those two, showing that it's not just black and white, there's some gray in between. And how do these cells develop? So you have the lymphoid arm and the myeloid arm, and all these come from the hematopoietic stem cell in the bone marrow. And these are the cells that then keep on differentiating. The lymphoid arm is the one that ends up giving us the T cells, B cells, and natural killer cells, which are also lymphocytes, and another newly discovered cell type called innate lymphoid cells. So these two are innate, but they are still lymphoid cells. So they still fall under that. And then the other arm is where you get everything else, like granulocytes, specifically eosinophils, neutrophils, and basophils. And then also that myeloid arm is the one that gives you monocytes and macrophages, which are closely related. Generally, macrophages are usually derived from monocytes in the body, but they are tissue-based, while monocytes will be circulating in blood. And then dendritic cells also, which are also derived generally from monocytes. But they are also tissue-based, and they have slightly different functions. So another thing that I wanted you to appreciate is initially, remember we said that innate cells are the ones that take place in the earliest immune responses because they are ready. They don't require any education. As they are ready to, they are fully equipped to fight any invader. While adaptive immune cells, like B cells and T cells, require some activation and some education so that they can produce an immune response which is tailor-made to fight the invader. So in between, you have macrophages and dendritic cells, which are components of the innate arm of the immune system. So those are the ones that act as a bridge between the innate immune system and the adaptive immune system. So when you think about it in terms of time, these two cell types, macrophages and dendritic cells, are the bridge. Because when the body starts an immune response, an innate immune response, because it has detected an infection, it will want to maximize on that. And instead of just letting the innate immune response die out and then starting an adaptive immune response totally independently, what the body does is to actually use the innate immune system to prime the adaptive immune system. So the innate immune system that is able to come very early is able to fight the infection on its own, but because it is not giving a tailor-made, highly educated immune response that is targeted for that particular pathogen, the innate immune system will not always be as efficient. And that's the reason why, as the innate immune response goes on, adaptive immune cells start getting activated so that they can come with a precise targeting of the invading pathogen. And so during that process of the innate immune response, macrophages, which are components of that innate immune response, will be busy activating B cells and T cells and providing some signals that educate those B cells and T cells so that the B cells and T cells can take over and eliminate the infection. So something that is quite important as far as adaptive immune cells are concerned is that they are able to create memory, immune memory. And that is part of the education. Once they get educated, in addition to the education, to refine the immune response so that it is targeted to that pathogen very precisely, they are also able to acquire features that enable them to remember that pathogen in future. And this is very important because this is the basis for vaccinology. And so that means that if you are ever exposed to COVID, or if you ever had COVID in 2020, if you will encounter COVID again in 2022, your body will not be starting from scratch. It will be having some memory, and then it can generate a more rapid and stronger adaptive immune response. So ordinarily, what usually happens is when you get your primary exposure, let's say you get exposed to COVID, it will take a while before the adaptive immune response comes in. And so in this case, we are looking at antibody levels. Remember, antibodies are produced by B cells, which are part of the adaptive immunity. So we can use antibodies as a proxy measure of the B cell response in a person. So once you get exposed to the pathogen like SARS-CoV-2, there'll be a lag period before your B cells get activated and start producing antibodies. In the meantime, innate immune responses will be dominating during this period. So any control of SARS-CoV-2 that you have during the very early days of infection, that control will be mediated by the innate immune system, like things like natural killer cells, neutrophils, soluble factors, those kinds of things. So then your B cells come in, they start producing antibodies, and that's what we call the primary response. But as you can see, that comes a bit late, and it's not very strong. It will be good enough, but yeah, it will have its own limitations. Once that pathogen has been cleared, because now you have very good T cells and very good B cells, the adaptive immune response will decline because there's no need of maintaining very high numbers of those cells in the body when you don't have an infection ongoing. But instead of declining to zero, you have a proportion that is left as memory cells. And these cells will be there in the long run. And even if you're exposed to the same pathogen 5 years later, at least you'll not be starting from scratch like you did in the primary response. It will be quick, and it will be stronger because you also have a larger starting number. So that's what a secondary response does. And how we take advantage of this in vaccinology is, instead of having a primary exposure, which is a real exposure to the pathogen, we expose you to a vaccine antigen, of which a vaccine's antigens will be components of the pathogen that is likely to infect you. So that means that the body will perceive as if you have had a primary exposure, and then it will generate primary response and keep memory. In the event you ever get exposed to the actual pathogen in future, that means that you'll have a better immune response. So that's the principle of vaccinology, and that's how we take advantage of how the adaptive immune system works. So far we have talked about so many cell types, and you might wonder, why do we have all this? The reason is that some of these cells are very specialized, that to an extent that whatever they do, it's very unlikely for any other cell to have the resources to do it. For instance, B cells are the only cells that produce antibodies in the body. So, and it will be extremely difficult for a biological system to come up with a cell that is able to do both that and do the function of CD8 T cells. So you have all those specialized cells, similar to what you see in our societies, where people specialize in different things. And then the thing is, these cells work as a community. You have cells like CD4 T cells that are coordinators of all the other components of the immune system. CD4 T cells will be there to coordinate the response of B cells by providing signals to activate B cells. Similarly, it will also provide similar signals to CD8 T cells. It will also provide some signals to macrophages, dendritic cells. So you find it in the middle, and it's the one that is coordinating all those responses. But even these other cells, you will also find them having interactions. Like dendritic cells will activate CD8 T cells while interacting with the CD4 T cell at the same time, giving you a triad there. So basically, you have a network of immune cells that are working together, basically forming what you would call a choir, and you expect every member of that choir to do their part so that the entire choir can give out the best melody. So that's how this works. So you'll find that if an immune system ends up having a defect in the CD8 T cells, some other components that are indirectly linked will be affected. So this is, and this diagram that we are showing here right now, the one we are seeing now, is an oversimplification of the network, the networking between the immune cells. So just to emphasize a little bit on that network, just to show just one pathway in that network, we have a component where things like antigen presenting cells like macrophages and dendritic cells are able to, we call them antigen presenting cells, and these cells are able to present antigen. They're able to capture a component of a pathogen, process it, and then present it in a palatable way, in a way that a CD4 T cell could understand, could interpret, could detect. Same way for the CD8 T cells, dendritic cells are able to do similar processing. So just trying to emphasize how these networks usually are required. So this is just an example of how antigen presentation, which is one of the components of those immune networks, how that usually works. And antigen presentation is usually done by every cell in the body. Every cell in the body is able to pick components of itself, process it, and present it on its surface, so that that cell can be recognized as healthy. So that is that's a different pathway. But then you have the other pathway, which takes components of pathogens, processes them, presents them on the surface, so that other immune cells can see that pathogen in a way that they are able to see, and then they can start responding. So for instance, so the cells that mediate that second process, which is meant to detect pathogens, those are cells that we call professional antigen presenting cells. And those are mainly dendritic cells, macrophages, and also B cells also are considered professional antigen presenting cells. So these cells, what they will do, let's say here you have a macrophage, and you have a pathogen here. Let's say this is a SARS-CoV-2 virus particle. It will be taken in by the macrophage into intracellular compartments, where it will be broken down into small pieces. And then that macrophage will present small pieces. You see, like these ones, it will present some of those small pieces on its surface. And then that's in a manner, in the context that CD8 T cells can then be able to recognize that small piece, a peptide. And so that's how CD8 T cells will get activated by macrophages and dendritic cells. Similarly, on the CD4 side, the same macrophage or dendritic cell can present another piece of that SARS-CoV-2 virus particle on its surface in what we call an MHC class II protein. And then that piece of the virus that is presented can be recognized by a CD4 T cell. And then when the CD4 T cell gets activated because it has recognized this peptide from the SARS-CoV-2, it is only the CD4 T cells that are specific for SARS-CoV-2 that will get activated. When they get activated, they then now start mediating their coordination of the immune system. So they'll go and activate a B cell, and that B cell will also be presenting a similar peptide on its surface from SARS-CoV-2. And that's how that CD4 T cells will be able to recognize, oh, this is a B cell that I need to activate. At the same time, those CD4 T cells will also produce soluble factors like interleukins that will further activate the CD8 T cells that was also activated in the other pathway through the MHC class I molecule. So this is just to make you appreciate the network nature of the immune response. So this association between a CD8 T cell, an antigen presenting cell, and a CD4 T cell, that's what I was presenting here as a triad that you can have. So, so this slide somehow shows the functions of CD4 T cells, because you can see the central role that the CD4 T cells are playing. But how do now? Let's say that now CD8 T cells have been activated. What next? This person is infected with SARS-CoV-2. The body was able, the dendritic cell or macrophages, they were able to detect them, especially dendritic cells, they were able to take in the SARS-CoV-2 virus particle, process it, present it on the surface, and therefore be able to activate CD8 T cells. And on the other side, at the end of the day, B cells were also activated. So then what next? Activation is just meant to prepare them for their effector functions. So CD8 T cells, once they get activated, this cell, we call them cytotoxic T lymphocytes, because their work is to kill infected cells. So if you have a cytotoxic, this is a CD8 T cell that has been activated through the process that we have just seen in the previous slide. What it does is to go looking for any cells that are presenting the same peptides. The cells that are infected with SARS-CoV-2, they'll go and scout for them. And once they recognize them, they'll then release some soluble factors and things like granzyme B and perforin, and soluble factors like cytokines. And they'll release those ones directed to the infected cell, and then those are the things that are going to kill the infected cell. So the granzyme B and perforin are usually released in what we call the cytotoxic granules, while you have the cytokines that will also be released, which are other soluble factors that will be able to restrict viral growth in the infected cells. And that's what will kill. And once you kill the infected cells, you are curtailing continued viral replication, and that's how CD8 T cells enable the body to control replication of viruses that have infected the individual. Then the B cells, we saw that the B cells also get activated at the end of the day. But once they get activated, what do? They produce antibodies. So you can have B cells that have been activated, and then they differentiate, they acquire features that enable them to secrete antibodies. And those antibodies have various functions. For instance, there are those that mainly work by blocking the target, the pathogen that is invading. So they just bind on the surface of the pathogen and therefore prevent the pathogen from interacting with its target cells. And that means that the pathogen has been neutralized. There are antibodies, sometimes also, okay, the same antibodies could also go and coat that pathogen and make it more palatable for other immune cells to come and take it up, phagocytose it, and destroy it. So that's what we call opsonophagocytosis. And then you also have antibody-dependent cellular cytotoxicity. This is where the antibodies that are produced against the pathogen, or even against the pathogen, will be able to identify cells that are infected by that pathogen, because those cells will be having proteins from that pathogen on their surface. And then they then bind to that cell that is infected, and then that will make that cell more palatable for attack by other components of the immune system, such as natural killer cells. So if you have a cell here, or even a pathogen, but mainly a cell that is infected, you'll have natural killer cells coming to identify it, because that infected cell will be coated by antibodies that were produced by the B cells. And so that will be very similar to what CD8 T cells did, whereby they just minimize viral replication by eliminating all infected cells. Then antibodies can also go and coat the pathogen, the target pathogen, and make it more easy for it to be targeted by other factors such as complement proteins. And that will enable the lysis of those pathogens, and it can also mediate inflammation, which is required. So all these functions are mediated by different components of an antibody. An antibody is usually a Y-shaped molecule, which is produced by B cells that have been activated. And then it has what we call the Fab region, which is the part that binds the pathogen directly, the targeted pathogen directly. And then you have another region, the tail end, which is called the FC region, which is the one that brings on board all these other components of the immune system once an antibody binds to its target. So this is the part that will become like an adapter that will link the pathogen, which is bound by the antibody, and things like phagocytic cells, like macrophages, or natural killer cells. So now let's just go a little bit deeper into the immune response to viruses. And we have covered most of this actually. So from this point onwards, it will be mainly revisiting those concepts, but with viruses in mind. So like, for instance, if you are exposed to SARS-CoV-2, you will have all the innate immune cells, many types of innate immune cells responding, recognizing that there is an invader, and therefore taking it up and destroying it by phagocytosis. And then on top of that, remember we said that some of these innate immune cells usually act as a bridge with the adaptive immune system. So you will have things like monocytes and macrophages and dendritic cells producing soluble factors that can modulate the immune response for T cells and B cells. And we have also seen antigen presentation, which is usually coming from antigen presenting cells like the dendritic cell. You'll have them presenting those antigens, the small pieces from SARS-CoV-2, to T cells, and then those T cells can start responding and they can start coordinating the immune response. And the T cells will respond differently depending on the subtype of T cell. Like CD4 T cells will produce more cytokines to coordinate the immune system in a more refined manner, while CD8 T cells will go and release cytolytic granules that will kill the infected cells. Well, some of those T cells, like the CD4 T cells, will interact with B cells and instruct the B cells to differentiate into versions that can produce antibodies. And then those antibodies can go and mediate these functions. So we can then move into HIV as an example. Any question before we move into HIV as an example? Okay, I see Mohammed asking about major histocompatibility complex. Yeah, so that is the MHC molecule that I showed earlier. Let me just go back. So these proteins that usually present those peptides, once peptides have been generated within the antigen presenting cell, they're usually loaded into this major histocompatibility complex molecules. And we have two types. The MHC, major histocompatibility complex, is the one that we abbreviate as MHC. So we have the type 1 and type 2. And the type 2 is the one that enables interaction with CD4 T cells, while type 1 is the one that enables activation of CD8 T cells. Is that clear? Okay. Then what is TH1 and TH17? Okay, I didn't want to go deep into that, but I'm glad you have noticed it, Mohammed. So yeah, TH1 and TH17. So amongst T cells, we usually have CD8 T cells and CD4 T cells. And CD4 T cells are the ones that are coordinators of the immune response. But within that, we have subtypes. And that is dependent on the kind of cytokines that they are able to produce. And also the kind of soluble factors, which we call the cytokines, that they are able to produce. So yeah, so the TH1 responses will produce T cells will produce things like interferon gamma, which is one of the cytokines that helps to activate the immune responses, some immune responses. And most viral infections are controlled by TH1 responses. And then you have others like TH17 responses, which will produce cytokines like IL-17, as the name suggests. And those are the ones that are quite involved also in viral infections, but quite a lot in bacterial infections. They tend to be more effective against bacterial infections. We also have what we call TH2. This will produce things like IL-4, interleukin-4, that is mainly quite effective in fighting parasitic infections. So you will find that there are subsets within the T cells, the CD4 T cells, and those are the ones that we call TH1, TH2, and TH17. Yeah, someone, Ian is asking how the CD8 T cells inhibit viral replication. I can go back to that before we proceed to the next phase. So when you have a CD8 T cell that has been activated through this process, this T cell will go and look for, okay, remember this T cell was activated because it saw a peptide on an MHC1 major histocompatibility molecule, one molecule. It saw a peptide, and this peptide was in agreement with the receptor that this T cell has. This T cell has what we call a T cell receptor. So the T cell receptor has to match with the piece that was presented so as to be able to recognize that, oh, yeah, I have this receptor, and my receptor is meant to recognize this peptide, meaning that I should swing into action, and then it gets activated, okay? So once that happens, this will be the CD8 T cell. We use CD8 and cytotoxic T cell interchangeably. So this will be the CD8 T cell. Now, cells that are infected by that virus will do a similar processing of particles of that virus. And then they will present them on their surface on an MHC class 1 molecule. So you will have the peptides the same peptide. And remember, this peptide then will be very similar to the one that the CD8 T cell saw when it was getting activated. So that's when it realized that, oh, this is the cognate peptide. And that means that this T cell here, no, this cell here, this infected cell here, is infected by the pathogen that I was activated to fight against. Basically, this cell is infected by the pathogen that the CD8 T cell has always waited for its entire life. And so then they form a synapse, and the CD8 T cell then releases these cytotoxic granules, which will then go and make holes in this infected cell and kill it and cause what we call apoptosis. Apoptosis is programmed cell death. So when the cell encounters these granzymes, and perforate, some of them will go and perforate, others will, then they activate programs that instruct the cell that it needs to die. So that's how apoptosis happens. And in the process, you have eliminated the factories that are churning out the viruses. So yeah, so the Th1 and Th17 are subsets of T cells that have been defined based on the cytokines that the cells are able to produce. And then I think I've explained what apoptosis is the programmed cell death. Yeah, I can also repeat a little bit about the antigen presenting cells. So basically antigen presenting cells will be members of the innate arm of the immune system, but they are the same ones that crosstalk with the adaptive immune system. And the main ones are macrophages and dendritic cells, and also B cells also play an antigen presenting role when they're trying to harness CD4 cell help. So what these antigen presenting cells will do is they take in the pathogen, they engulf it, they take it into some compartments called endosomes, which will merge with enzymatic compartments that are carrying enzymes, and then that pathogen is digested into pieces, into peptides. Remember, it had proteins, now it is broken down into peptides. And then those are the peptides that are loaded into the MHC molecules to mediate activation, to enable interaction between that antigen presenting cell and CD8 T cells or CD4 T cells. And that's how those CD4 and CD8 T cells are activated. So someone else has asked, inhibitory secretions produced by Th1 cells are. So things like, if I give you an example, these cells, yeah, the different subsets of T cells will produce different kinds of cytokines, and you can see CDS will produce some cytokines as well, and also some cytotoxic granules. So Th1 T cells will produce cytokines like interferon gamma. One of the things that we know about interferon gamma is that it limits how do I put it? Yeah, it limits things like mycobacterial growth. So if you have a cell that is infected by a bacteria, like TB, that's the growth of TB in that cell. If that cell is exposed to interferon gamma that is coming from this kind of cells, that TB will not grow efficiently. For viruses, we have something like interferon gamma also will if you have another cell like a macrophage which is infected with HIV, for instance, and this is exposed to interferon gamma, that interferon gamma will induce components, other pathways. It will activate pathways that will limit production of HIV from that infected cell. So that's just one of the examples of inhibitory cytokines that are able to inhibit viral growth or bacterial growth. I see Kamunya asking granulocyte colon. Oh, okay. Yeah, it's basically, Kamunya is basically talking about the full name of that cytokine, that soluble factor that can be produced by some of those cells. Onyango asking granulocyte monocyte colony stimulating factor, how that differs from IL-17. Yeah, every cytokine is different from the other. What is emerging is that IL-17 will activate programs that will fight bacterial infections mainly, while GM-CSF is a bit more generalized. It's also targeting bacterial infections, but slightly differently. And the other thing is that these cytokines also sometimes they have overlapping roles, and therefore it's a very murky world in there trying to. There are those cytokines that we know their exact role. There are some cytokines that are inhibitory. Some of them actually dampen the immune system, while others will activate effector cells that are meant to control the infection. Others are there to activate other immune cells so that those immune cells can infect those cells. So it's a very complicated network. So you cannot you cannot always compare and ask this cytokine versus this cytokine. Sometimes it's clear cut, but sometimes it's not as clear cut. Things like IL-17, we are only starting to understand them now because we are starting to appreciate that they might be important for controlling things like TB. But as far as HIV is concerned, in as much as those cytokines are produced when someone is infected with HIV, their role is not really very well known. It could be something like maintaining the integrity of some other cells, but we don't know. So it's quite murky there. That's a very good question, but just keep it in mind that there are those cytokines that, like the ones that I've given you, that we really know their roles very well. There are others that it will depend on who you are listening to, because maybe they are interested in a particular cytokine for a particular disease, and therefore they will tell you that this cytokine does this and this. But you meet another person who is working on another disease, who is researching on another disease, and they have seen that cytokine mediating a totally different role. So yeah, I see. So the mod T cells attack is only by apoptosis and is to activate B cells only? No. Remember we talked about T cells that activate B cells. T cells, if you look at this, you can see that CD4 T cells are interacting with macrophages. They are modulating the role of the functioning of macrophages, of dendritic cells, of CD8 T cells, of B cells. So you can see CD4 T cells playing all those roles. You can see CD8 T cells playing a role where they are getting some signals from CD4 T cells, they are getting some signals from CD8 T cells, and they also mediate some reverse signals in the opposite direction. At the same time, they are also fighting the infection. So you cannot easily exhaust the roles of these cells fully, but what you can do is to identify the main role. And so the main role of CD8 T cells is to kill target infected cells, while the main role of CD4 T cells is to orchestrate that immune response. While the main role of B cells is to produce antibodies. And the main role of macrophages is and dendritic cells is to do antigen presentation. So that's what I want you to take home. That's a simplified version. I'm going to answer 4 more questions. Let me see. I can see someone saying TNF alpha inhibits human immune system and promotes tumor growth. I don't know about TNF alpha being able to do that, Aaron, but I know about IL-10, interleukin-10, being able to do that. Which other one? TGF, tumor growth factor, not TNF. TGF beta is able to inhibit the immune system and allow tumor growth. There are also other factors, there are also other things that are not necessarily soluble factors that are able to inhibit immune system and allow things, the growth of tumors and such. Also even allow the continued replication of a virus. Like for instance, in IL-10, you have some cells that will produce, in HIV, you'll have some cells that will produce IL-10, and therefore they are dampening the immune response. And the virus can take advantage of that so that it can propagate itself. But also, some of those components that inhibit other components of the immune system are important in preventing autoimmune responses, autoimmune diseases. Because autoimmune diseases usually occur when some of those checkpoints are eliminated. Yeah, I see Kamunya clarified about CD40 cells. Yeah. Kelvin talking about IL-4. IL-4 is just one of the many interleukins. Yeah, IL-4 is produced mostly by Th2 T cells. Someone is saying is confused. Kelvis, you can always drop your question down there and then we can answer it at the end. Do CD4s attack only microbial infected cells? Or I think you are referring to CD8s. Do they only attack microbial infected cells? Yes. If they start attacking healthy cells, that's when you end up with autoimmune diseases. Which specific CD50 cells and IL factors are a must know in terms of their role in the disease progression? Oh yeah, I think Joroge, I clarified that, the main things that you need to take home. Yes. So we can then proceed to HIV. So you know about HIV, all of you, and this is just some of the statistics that we had as at 2019. I will need to update this to 2022. And yeah, so we have around 38 million people globally who are living with HIV, and 25 million are on ARTs, and we are getting 1.7 million every year new infections, and we are still getting around more than 500,000 people who are dying every year. So yeah, we have had around 75 million people getting infected and around more than 30 million who have died. And this is a lentivirus that belongs to the retroviruses family. And it has many proteins. And these are the proteins that we take advantage of when we are trying to fight this virus. So you have we have the genetic material, the RNA. You remember this is an RNA virus. We have 2 strands of RNA in the middle. And then we have some enzymes that are important for the survival of this virus, things like protease, integrase, reverse transcriptase. Those ones are also components of the virus. We have structural proteins like P24 capsule proteins, which mainly provide the structure to accommodate the enzymes and the genetic material. And then you also have what we call the matrix proteins. And you have the envelope. This envelope is usually derived from the cell that produced this virus. As the virus buds out, it goes with a piece of that cell membrane from that infected cell, from that cell. So the virus goes with part of that. But then in that envelope, it has inserted things like proteins, other proteins, which we call them envelope proteins, such as GP120 and GP41. So most of the vaccine efforts, as we speak, are targeting these two because they are the ones on the surface, the GP120 and GP41. We also have some components that, some kinds of immune responses that are not dependent on the protein being on the surface. And those ones target mainly things like the P24 and the matrix. Specifically, for antibodies to neutralize this virus, they need to bind on something that they can access. So antibodies that we try to elicit using vaccines are meant to target GP120 and GP41. Whereas T-cell responses, remember T-cell responses, eh? I hope you guys remember how I explained this. Remember, T-cell responses are driven by a peptide, which is derived after degrading the pathogen. So that means that a T-cell can see a peptide which comes from a protein, which is hidden inside the virus. So while an antibody, like here, can only bind on things that are on the surface. So because of that difference, you'll find that even these internal structures, they can be, when the virus gets ingested by an antigen-presenting cell, and the whole virus is degraded, peptides that are coming from any part of this virus, including the ones that are hidden, will be able to activate a T-cell response. So when we try to induce T-cell responses with vaccines, we want those T-cells to recognize peptides from both the surface envelope protein and also from the structural proteins that are hidden within, because T-cells will be able to see peptides that have been generated after the virus has been degraded by antigen-presenting cells. But for antibody responses, we will want to elicit antibodies that target the envelope proteins. So all these are mediated by 9 genes, and we have the 3 main genes, which are structural or enzymatic genes for enzymes. Those are the main ones. The GAG is mainly for structural proteins, and the envelope also is for structural proteins. The envelope, of course, it will be the GP120 and GP41. Then you have the polymerase gene, the Pol gene, which will have enzymes like the protease, the integrase, the reverse transcriptase. So that's the Pol gene. And then you have others that are called regulatory and accessory genes. So HIV is a very clever virus. It has, along the way, acquired some of these genes that will enable it to evade the immune system in one way or the other. So for instance, some of these things like NEF will go to T cells, and when T cells get exposed to NEF, they downregulate some of the surface molecules, which are important for their function. And that's one of the things that the virus will use to dampen the T cell response. So these accessory and regulatory proteins are quite important for HIV survival. And sometimes we also target them. But our main targets with vaccines are these main ones. So what happens when someone gets infected with HIV? I'm sure you all know about the CD4 T cell depletion, because HIV mainly infects CD4 T cells. And remember, the T cells play a very central role here. So if you have HIV coming to eliminate these ones, it means that other components of the immune system are likely to collapse. Remember, I talked about an immune orchestra or a choir, and CD4 T cells are like the conductor. When the conductor is not there, the choir starts falling apart. So that's what HIV does. It will target those CD4 T cells. And so here I'm showing you a graph, a cartoon of a graph. You probably have seen this one as well, which is showing the CD4 T cell counts in blue and in the blue line, and the viral loads over time in red. And this is from the early times after infection all the way into the chronic phase of HIV infection. Because remember, HIV infection can last many years. And so we have what we usually call the acute phase of HIV infection. This is usually within the first 3 months at most. Most of the events in the acute phase usually happen in the first 30 days, the first 1 month. But generally, we can extend that period to talk about the first 3 months and talk about acute and early infection. And then you have what we call the chronic phase, during which most people are fairly asymptomatic. This will therefore say that it is a period of clinical latency. And then we have the end stage disease stage, where someone is developing AIDS and opportunistic infections start coming up. because HIV has just knocked out all the CD4s and therefore in the process knocked out other immune responses and allowed every other disease to come up as an opportunistic disease. So during primary infection, which is during the acute infection, you get sharp decline in CD4 T cells. This is even within a week after detection of virus in peripheral blood, which will be probably around 2 weeks. We still don't know the exact period, how long it takes for the virus to get detectable in blood, but estimates have been around a week or two. So then you start detecting the virus in peripheral circulation, and then you start seeing sharp decline in CD4 counts. During that same process time, you have a sharp increase in viral loads because now you're able to detect the viral loads. And the reason why there's this very rapid increase in viral loads for the virus is because the immune response is ill-prepared, is not prepared to fight this virus. So at this time, you are getting the only components of the immune system that are participating are the innate immune system, which is not very efficient. And that's the reason why the virus is really thriving. And then after some time, you start getting adaptive immune responses like antibodies and CD8 T cells getting activated, getting educated, and then they start controlling viremia. But then the problem with HIV is very few people will be able to control viremia to below detection. But the majority of the people, 99% or so, will reach a point where the rate of viral replication and the rate at which the immune system is suppressing that viral replication strike a balance. And that's what we call the viral set point, and which maintains some level of stability as the person moves into the clinical latency stage, the chronic phase. But then later on, the viral because the immune system has degraded so much, the viral replication wins, and then that's when you see increases in viral loads during the end stage disease. So during that time, of course, you have this sharp decline in CD4 counts during acute infection. And once the viral replication has been partially controlled, there's partial recovery of CD4 counts, but as the person moves into the chronic phase, there's some gradual decline of CD4 T cells that happens in the background, in as much as this person doesn't have clinical symptoms. And when that reaches a very low level, that means that also other components of the immune system have been compromised. And then that's when we end up with the end stage disease. So yeah, I just decided to show you some real data because I mean, what I've just shown you here is a cartoon, just so that you can actually see that that trend is actually what usually happens. But this data shows the viral load kinetics in the first 1 year in people who were followed very closely, and unfortunately, a few of them got infected, and they were able to be monitored during that first year. This is historical data which was generated when people were not able to be started on ARVs immediately, and their CD4 counts needed to decline before they could be initiated on ARVs. Those are the ones that I'm showing on the left, which we are calling untreated. So you can see that in these people, viral load, and each of these lines and colors is representing one person. So you can actually easily follow how the viral loads change within like the green person over time. So you can see that during those first few days, up to around 30 days, there was rapid increase in viral loads. And then it reached the peak, and then it started coming down. And then for most people, they established a viral set point before 50 days. And then in the course of that first year, their viral loads were fairly stable. And that's what we call viral load set point. So that is for untreated people. And you can see that the CD4 counts here at the bottom, I'm showing the CD4 counts. You can see that they started with the CD4 counts that were on average around 1,000, and then there was a sharp decline for almost all of them, and partial recovery and as they moved into the chronic phase. So this is really real data from individuals. But then the policies were changed, and now the researchers who are collecting this data were allowed to treat people immediately when these people get infected. And so you can see that in this group of people who are later on infected, when the viral load was detected, they were put on ARVs immediately, and the viral loads became undetectable very fast. So when people are treated early, you are able to knock down the viral replication to below detection. Unlike people who are not treated early, where the virus remains detectable, only that it is at that set point, but it's still detectable and the immune system is still degrading. And so you can see in agreement with all that, the CD4 counts did not really, when you start people on ARVs very early, the CD4 counts did not really decline. They remained fairly stable, unlike here where they declined and only had partial recovery. This group that was treated early maintained fairly good CD4 counts. Yeah, so this I had gotten this data from a paper, a publication where I was measuring cytokines at all these different time points. That's why we have these cytokine determinations here, so you can ignore that part. So you have seen how real data looks. Now let's come back to the simplified cartoon. So during that acute phase, during that acute phase of the infection, do you remember what I talked about, which kinds of immune responses will take place during the acute phase? The very, very early days. Anyone? What kind of immune cells, which kind of immune cells would you expect to participate during this phase, the acute phase? Innate immunity. Great, we are together. Hey, you guys are very awake. I'm very impressed. So, yeah, during that early phase, you expect mainly innate cells to participate. Things like monocytes, natural killer cells. You'll also expect, you remember those cells that we said that are in between? They have features of both innate and adaptive immune cells, like natural killer T cells. We'll also have them getting activated during those early phases. And one of the things that these cells will do is to produce a lot of cytokines, because these cells are not really educated to target the infection in a very specific way. They usually just produce soluble factors that are meant to just make the body inconducive for any pathogen that is invading. So you end up with a lot of proteins, soluble proteins that will be produced. Some of them fall into that category that we call cytokines. And this is what we usually call the cytokine storm. So in HIV, a few days after someone is infected, they are likely to get flu-like symptoms because they are just loaded with a lot of cytokines that are causing a lot of inflammation and causing a lot of chaos in the body. But the body is indeed trying to just make the environment unconducive for the invading virus. So that's what usually happens during acute phase. And innate immune cells and also a lot of cytokines that will come from those innate immune cells and some proteins that will try to control the infection. But the infection will continue to grow anyway because it is not very efficient. The innate immune system is not the most efficient. But then you have CD8 T cells starting to kick in, which are adaptive immune cells that are very targeted. And it has been quite interesting in HIV. The emergence of CD8 T cells coincides with the starting decline of the viral loads. This has really pointed to the possibility of CD8 T cells being the major contributors of this initial decline. And that has led us to invest a lot of efforts in understanding how we can reduce the best CD8 T cell responses. But the HIV virus is very clever. What it will do is it will change small portions of those proteins that it has. And then the CD8 T cells will not be able to recognize it anymore. And then the body has to elicit new CD8 T cells that can recognize the new variants. And that's what we call the mutation and virus escape. So that's how we think that is what curtails this decline to some extent. And then you end up with that set point where you have an arms race between the immune system and the virus. The virus escapes the current immune responses. The body produces immune responses that are targeting the new variants that have been generated. And it continues like that. And we still don't really understand whether CD8 T cells control viral replication in chronic infections, but there are some indications that happens, but it is not very well defined. So the best thing is to assume that CD8 T cells are also important in controlling viral replication in the chronic phases. So then the other arm of the immune response is the antibody responses. So at around the peak, you also start seeing antibody complexes. And then these are antibody virus complexes. And it's because there's a lot of virus and very little antibody. It means that all the antibodies that are being produced are going to bind to the virus. But then as the antibodies accumulate in the system, you start seeing free antibodies. And these are targeting things like GP41, which is one of those envelope glycoproteins. And so you get some neutralization after some time. Some good antibodies that are able to block the virus are generated. But then the virus also starts escaping from those antibodies. But what happens is those antibodies are targeting that virus that is infected that particular individual. And we call those antibodies autologous and neutralizing antibodies. But then over time, as the virus keeps on escaping and the body adjusting the antibodies, we end up with some immune response that is able to cover many viruses. And in a few participants, that is able to broadly cover all viruses or the majority of the viruses that circulate in the world by targeting some regions that the virus is unable to mutate. Because if it mutates those regions, it becomes less fit. Those are what we call conserved epitopes. So it's, in a few people, you end up developing antibodies that target conserved epitopes, and those are the ones that will be called pro-neutralizing antibodies. And those are the antibodies that we want to induce with vaccines, because it means that if someone has pro-neutralizing antibodies before they get exposed to HIV, they are likely to block any HIV that they are getting exposed to. So that's our vaccine. That's where our vaccine efforts, a lot of our vaccine efforts are going into. In fact, the vaccine efforts that we are doing at CAVI and the projects that I'm working in are aimed to induce pro-neutralizing antibodies. So we can just revisit CD4 T cells again a little bit. You remember that they are the coordinators of various components of the immune system, and HIV, what will do is it will knock down those ones, you can see, and that's what causes the immune system to collapse eventually. So at the end of the day, what we want to do is to take advantage of this knowledge and generate interventions. And one of the main interventions that we have wanted to generate is vaccines. But we can also use the immune system to do some direct therapeutic interventions, which are alternatives to antiretroviral therapy. So what sometimes we do is we go and pull out the antibodies that people who are HIV infected are producing. We look for the genetic material. We extract the genetic material that encodes for those antibodies, and then we are able to produce those antibodies as recombinant proteins in the lab. And then once that happens, we can use those antibodies to inform vaccine design, because if that antibody is a pro-neutralizing antibody, it means that you can work backwards using what we call reverse vaccinology to design antigens that can elicit an antibody that is similar to that, or antibodies that are similar to that particular one. At the same time, we can use the same antibodies which are produced in the lab. We can purify them, ensure that they are clinical grade monoclonal antibodies, and then we go and infuse them in people who are HIV infected so that those antibodies can suppress viral replication. So that means that those antibodies will then actually be used for therapeutic purposes, similar to ARVs, while at the same time also informing vaccine design so that we can have vaccines that can guide the body for the body itself to produce similar antibodies. So that's one of the, those are two of the main two of the main applications of our immunology knowledge in combating HIV. So you can do passive immunization by administering those broadly neutralizing antibodies as infusions. And then that means that if that person is HIV infected and you have given them a cocktail of those antibodies can control viral replication, independent of whether that person is taking ARVs or not. And if they are on ARVs, it means that they can actually stop taking ARVs, and the antibodies that you have infused in them will be able to control virus replication, and that person doesn't need ARVs. So, but the problem is when you do that, these antibodies decay over time. Because remember, these are antibodies that have been produced outside the body, and they will decay over time. So that means that later we will have to keep on giving infusions. Instead of someone taking ARVs, every 2 or 3 months, they come back for an infusion, and then their viral loads are controlled. So that is something that is in development. It is not yet licensed to be in the clinic, but it is one of the things that might be applicable in the future. And then in addition to that, these broadly neutralizing antibodies, if you infuse them into someone who is exposed to HIV, let's say this person is like a discordant couple, where one person is not infected and the other one is infected, it means that those antibodies can protect the uninfected partner, at least for the duration of time when they have not decayed below levels that are required, below the threshold. So that's one way that we use these monoclonal antibodies that we have collected, or rather synthesized in the lab using information that we have obtained from HIV-infected individuals. And of course, now the advantage of the other strategy, the vaccines, is that if you work backwards from that broadly neutralizing antibodies to determine which kind of protein can elicit them, can instruct the immune system to elicit them, that means that you can just administer that antigen to the person, and the immune system of that person will generate antibodies against that antigen, or also T cells, depending on the immune cells that are participating in that immune response. And the advantage of that is that those cells are cells that keep memory. So that means that in the event that that person gets exposed to HIV in future, those T cells and those B cells can go back and produce more immune responses. And that's how a vaccine works. Remember, vaccinology is pegged on the ability to induce immune memory. So that is a more sustainable strategy. And we have some vaccines that are in the pipeline right now, but I'm just going to show you some of the attempts that we have done for the two strategies, in as much as they have not been licensed as yet, to show how far the field has come for those two strategies. So this is a study that was done in South Africa. And in this study, this group of scientists, they took one of those broadly neutralizing antibodies. It's called VRC01, infused it in people who are highly exposed to HIV. These are commercial sex workers who they'll receive all the counseling that they need to practice safe sex. But these are people whose profession, being commercial sex workers, will generally expose them to HIV. And so they were infused. Some of them were infused with these antibodies. And it was shown that those people who had received these antibodies were protected from viruses that could be neutralized by this antibody. So in a way, showing that these antibodies can be used to protect, very similar to pre-exposure prophylaxis that is being done by giving people ARVs, people who are highly exposed to HIV being given ARVs so that they can continue being protected by the ARVs from acquiring HIV. So these antibodies, it was shown that they can actually protect if the viruses are sensitive. So the idea is to ensure that you use a cocktail of antibodies so that all viruses out there are covered, and then the person will be protected. And then here we have the several HIV vaccines that we have had in the past. And this started as early as 2005, 2004. The earlier vaccines that were tried, they were not protective, like VAX 003, VAX 004. Then we had another one which was meant to induce T cells. These ones were meant to induce antibodies, the ones that were there at the very beginning. Then we switched to T cells because of the frustrations from the antibodies at that time. There was no protection. Then another T cell, a vaccine that was tailored to induce T cells, no protection. Then there was some light at the end of the tunnel in one of the vaccine trials that was done in Thailand. And this was meant to induce antibodies. It was an improvement of these earlier vaccines, and it showed 31 efficacy. This was super, super exciting to the field. Because I mean, this was happening almost 30 years from the time when HIV was discovered. And it was the first time there was a positive signal. However, it was very low. That positive signal was very low, and the vaccine could not be licensed. So what did we do? The field went and modified this so that the vaccine can suit viruses that are circulating in Africa. And this is what was tried in South Africa and several other African countries. No protection. It was quite disappointing, no protection. So you can see that it has been quite a struggle as far as getting HIV vaccines are concerned. But right now we have some vaccines that are still very early phase. These ones that I'm showing here are the late phase trials where you're looking at whether they are protective. But we have very early phase vaccines that are only starting to reach the clinical trials right now. And we are only looking at their safety at this stage and whether they are inducing immune responses. Whether those immune responses will be protective or not, we don't know yet. But these vaccines have been designed using all the immunology that we have learned from HIV over those 30 years, 30 or almost 40 years. So and because they are what we call rationally designed vaccines, whereby you look at a broad neutralizing antibody and you work backwards to find out which antigen can produce that. And when you test it in animal models, you can see that it's actually able to lead in that direction. So those new vaccines that are not included in this list because they are still early phase vaccines will probably give us a different story in a couple of years' time, maybe in a decade or so. So that's the end. I can see there are many questions. Let me see. So for concordant couples, what causes the paradox? I think here you meant discordant couples, Bill. Yeah, so for discordant couples, it is common in HIV to find that one person is infected and another one is not infected. And these are people who are actively involved in sexual contact. The transmission for HIV usually has a bottleneck. And it is not always a given that when two people are sexually involved with each other and one is infected, that the other one will acquire HIV. There are always chances that one person, the transmission will not occur. So that's what leads to discordant couples, because transmission of HIV is not 100% efficient. Unlike viruses like hepatitis B, which is very efficient in transmission if someone is not vaccinated and they get exposed. So HIV is less efficient, and that leads to that. However, there has been a lot of interest to try and see whether couples, when you have discordant couples, does the person who is not HIV infected, do they have special immune responses, like innate immune responses that protect them? The evidence from that has been a bit shaky, but it cannot be ruled out based on that, based on how logical it sounds. But the data sometimes supports that, sometimes doesn't. So, yeah. But generally, HIV is just not efficient, the most efficient in transmission. PrEP is when one is infected while one is not infected. Okay, I think here you meant PrEP, meaning the pre-exposure prophylaxis. I've talked about it. where if one person is infected and another one is not infected, the one who is not infected can be given ARVs to protect themselves from acquiring HIV from the one who is infected. And as I've mentioned, we are looking into ways of using broad neutralizing antibodies as a form of PrEP, long-term PrEP. There are also some groups that are also developing ARVs that are formulated in a long-term slow release formulation, so that they can also give long-term PrEP. So I hope I've clarified that. No, discordant couples, they don't both. Oh, if you mean concordant couples, yes. But when you talk about discordant couples, yeah, one of them is infected, the other one is not. PEP, you have the other version called PEP, post-exposure prophylaxis, where if someone has been exposed to HIV, they can get some ARVs, and in case HIV was trying to infect that person, that infection process can be curtailed. And antibodies are also being evaluated also in some studies to see whether they can be used as a form of PEP. In case someone is exposed, within a small window period, they can go and get an infusion of those broad neutralizing antibodies and then get protected from acquiring HIV, especially things like occupational exposures, those kinds of things. So PEP, both PEP and PrEP might benefit from having antibody versions coming into the clinic in the near future. PEP does not reduce viral loads. PEP actually protects someone who has already been exposed from acquiring the disease. If the CD4 cells are the ones that are affected by HIV and the CD4 cells are the ones that bring about the action of CD8 T cells, how do the CD8 T cells cause the viral load to decrease from the graph you showed? So it's because, so let me just go back. Yeah, so you have CD8 T cells coming here. But remember, this person, they still have some CD4 T cells. If you look at this graph, I mean, at that stage, the CD4 T cells have declined, but they are not fully wiped out. So that's the reason why CD4 T cells might be able to kick in here, and start mediating some suppression. But because of this decline of CD4 T cells, in as much as they are still there, we still think that might be contributing to the inability of CD8 T cells to knock this viral replication 100%. So it's because, yeah, in as much as someone is infected, they still have CD4 T cells that are there, as you can see in the graph, and those ones will, the little that is left will still be able to mediate, activate and coordinate CD8 responses. Can we get a cure by targeting the enzymes? Are you referring to the enzymes that HIV has? Let's see. Some of these enzymes, these are actually, that's what ARVs do. ARVs target the reverse transcriptase enzyme, others target the protease and the integrase. So yes, we have, it's not a cure, but we have a treatment by targeting enzymes on HIV. English Kuba Bill. Okay, Bill used a very, very complicated English. Okay. Can broad neutralizing antibodies be used as post-exposure prophylaxis medication? Yes, I talked about that. Discordant couples will use PrEP. Yes, we have talked about that. Yeah, the uninfected person is the one who will use PrEP. Is HIV receiving the same attention as COVID did? HIV has received way more attention over the years. You can imagine we have worked on this since mid-80s, when some of us were not born. Mid-90s, when some of you are still not born. So all the cumulative knowledge that has been accrued, all the knowledge that has been accrued over the years by studying HIV is the knowledge that we used to create treatments and vaccines against COVID very quickly. And the technologies that have actually been developed to try and fight HIV are the ones that are enabling us to fight other infections, emerging infections, very quickly. So COVID has not received as much attention as HIV. Actually, HIV is the one that has received more over the many years. But because the virus is very tricky, it mutates very fast, evades the immune system, and there are so many variants out there. That's the reason why we are stuck with it. We have not been able to have a vaccine as yet. I think someone said, yeah, I think Joy, I think you have clarified it there quite well. PEP is after exposure, while PrEP is for people who expect to be exposed. Yes, I see Nanga has clarified a little bit more. Bill, we don't know what, as I said, we still don't understand discordant couples quite well. We have our own hypothesis that they could have some special innate immune responses that could be protecting them. It could be just by chance. Yeah, there are some studies that have suggested that some people lack some receptors, like the CCR5 receptor, and those people are more likely to remain as discordant couples, as the negative partner in a discordant couple. So you are right, they might be lacking a factor that is important for them to acquire infection. So there are many possibilities. The possibilities are as many as our imagination. There is a book that we use for immunology. I will forward the title of the book to your class rep, so that they can forward to you. But it's more about general immunology, because as you have seen, most of the concepts that I've shown you here are things that are emerging. They're evolving. So these are things that we mainly acquire from publications and from our own research. So they are not yet in textbooks, mostly. But there are components of the immunology knowledge that is included here that are in textbooks. So I'll see if I can find one that you can refer to. Is it overly impossible to come up with a cure for a virus, or we can come up with one with advancement in biotech? There is a lot of effort that is going into trying to get a cure for HIV. Okay, there's something here that I didn't cover. Maybe I will include it for future classes, for the other lots. HIV goes into what we call reservoirs. So even when someone is on ARVs, you still have a few cells that are infected by HIV and are hidden in various places in the body. And so that's the reason why you cannot say that someone is fully cured. What you can do is you can control viral replication, or you can even confer what we call functional cure. Functional cure is whereby you are not fully cured. You still have some of those hidden viruses in the body, but your immune system is able to suppress any virus that tries to rebound. So that's where we are with the cure efforts. Yeah, so I'll try to see whether I can get some of those books that you can refer to. Yeah, I can see a lot of discussion on discordant couples. Yeah, sometimes babies who are born by infected mothers, sometimes they get infected, sometimes they don't get infected. And that's what we call vertical transmission, mother-to-child transmission of HIV. And we have programs that are meant to administer ARVs to HIV-infected mothers to knock down their viral loads and therefore reduce the chances of them transmitting to the mother, to the child. And also the child immediately after birth can also get some ARVs. Yeah, so it's easy to have, as we speak right now, we have very good systems of ensuring that a mother does not transmit HIV to the unborn or newly born baby. Even before those programs came in place, if a mother is not treated, there are chances that some babies will be born when they are not infected, but that proportion is definitely lower compared to now when we have those programs for preventing mother-to-child transmission. So it's somehow similar scenario. Yeah, I see Kamunya talking about genoise immunobiology. Actually, that's the book that I was thinking of forwarding to you. If you can actually get it, it will be a very good reference book for immunology. And then sometimes clinical trials can go wrong, but that is usually when the correct measures are not taken in place. Because I mean, by the time you have this new product, let's say you have this vaccine, and you want to try it in humans. By the time you do that, you have to test it in mice, then in large animal models like monkeys, or those kinds of things. And then before that, of course, you've done a lot of experiments in the lab. And then after that, you try it in adults who are healthy. That's what we call a phase 1 clinical trial. And you monitor them very closely in an hospital setting. So it is very rare. We have so many clinical trials that are happening and we don't have catastrophes taking place in those clinical trials. And it's because there are so many measures that are put in place to ensure safety of the patients. But we have had a few historically that have led to serious adverse effects. And we would really want to at least try to minimize that. In CAVI, we have never had that because we are always putting in place all those measures. Someone is asking about the Berlin patient. The Berlin patient was an HIV-infected patient who was cured, and basically they went and wiped out the entire bone marrow and the immune system, because this person also had a cancer. I think that person was a lymphoma. So they were trying to wipe away the immune system, and in the process, of course, they were also wiping away infected cells. And then they gave that person a bone marrow transplant from a person who lacks some of the receptors that HIV requires. So that means that the new bone marrow and the new immune system that this person had, the Berlin patient had, was somehow resistant to HIV because it was lacking one of those receptors. So that is one of the few patients who have been cured. So are we expected to read updates of the upcoming research on immunology and as far as exams are concerned, or just for our own personal knowledge? That is for your own personal knowledge. I don't expect you to go beyond this, whatever we have covered today. I believe whatever we have covered today is a good foundation for you. Then you can then go and explore. Because I mean, we are just trying to give you the power to read. Isn't it, Njoroge? So this is your starter pack for you to be able to read immunology on your own. Sometimes the ARVs can fail to work if the virus has escaped, because the virus is always trying to escape. So that's when someone is then initiated on a new regimen that virus is sensitive to. How much you are paid to be done the experiments? No, people are not paid. We don't pay people to participate in clinical trials. It's meant to be voluntary, and but there are some benefits that the person will accrue. But it's just you cannot buy someone's safety. There's no amount of money you can pay them. But what we will do is to minimize the inconvenience. And part of that is like, if you have a job and you missed going to work that day, you will be given back your fare. You can be given a compensation for the day's wage because you are not able to earn a salary on that day. And then we have medical cover for the clinical trial in case there's an adverse event, which rarely happens. that will take care of your cover of your medical needs immediately. And then participating in a clinical trial, that means that you'll be able to get medical checkups free of charge, because part of the things that we'll be doing in that clinical trial is to ensure that you are well, and therefore we'll be able to do free medical checkups. And there are some other indirect benefits that you'll get. But the main benefit is contributing to knowledge. And what we usually ensure as scientists is to ensure that you are safe. We are doing it in a way that is safe for you. No, there's no risk allowance. But what we do is we minimize the risk by ensuring that there's a medical cover for that clinical trial. Yeah. I can see you guys are quite excited about this. Yeah. So it's good to understand these things so that you saw what happened when COVID came. Everyone was asking you, I mean, you are a medical student, you need to explain to us this. So I'm glad that you guys will be able to handle those questions that are coming from the community, because we are the elites as far as medical knowledge is concerned in our community. So I don't have anything else. You guys enjoy your Friday, and thanks for being a very engaged class.